Tropical coral reefs are one of the most diverse, complex, and productive ecosystems in the world. Scientists estimate that the number of plant and animal species inhabiting coral reefs ranges anywhere from 600,000 to over 9 million worldwide. Many countries depend on coral reefs for their ecosystem services, including food production, coastal protection, and intrinsic values of biodiversity that generate tourism dollars from activities like fishing, snorkeling, and scuba diving. Coral reefs are one of the few ecosystems that can actually grow their own substrate. Scleractinian corals are hard corals, and calcifying algae lay down calcium carbonate, or limestone skeletons that grow and expand the substrate over time, making the perfect habitat for many species to colonize and thrive. The reef is full of complex interactions and dynamics between the species that live there. One interaction of great importance is that of corals and algae. Did you know? Worldwide, about 500 million people have some level of dependence upon coral reefs. When it comes to corals and algae, there are good interactions and not so good interactions. An example of a good interaction is the mutually symbiotic relationship between hard corals and the unicellular microscopic algae called zooxanthellae. The living animal of a coral is called the coral polyp. Thousands of these polyps are connected by a thin layer of tissue that allows for the sharing of nutrients throughout the entire coral colony. Inside this tissue lies the zooxanthellae, giving the coral its beautiful colors. The coral and algae each benefit from one another. The coral provides the algae with shelter and substrate to live on, while the algae provides the coral with delicious and nutritious carbohydrates and other materials produced by photosynthesis. This relationship is crucial to the survival and growth of the coral colony. Without the algae, it is unlikely that the coral colony will survive. Because of this photosynthesizing algae, corals are restricted to a thin layer of geographic distribution. Corals require a very stable environment with little changes throughout the years. Coral reefs are found about 30 degrees north and 30 degrees south of the equator, with some exceptions. These waters are relatively stable in temperatures ranging from 23 degrees Celsius to 29 degrees Celsius throughout the year. Coral reefs thrive in oligotrophic, nutrient-poor environments where sunlight can penetrate the water column and provide the photosynthesizing algae with plenty of light. Not all algae is beneficial for coral reefs, however. Macroalgae and hard corals compete for space on the reef. Macroalgae grows faster than hard corals, and in some cases the macroalgae can smother the coral, blocking it of sunlight and possibly causing mortality to the entire colony. Some algae have been shown to release allelopathic chemicals that are correlated with outbreaks of coral disease within the colony. Macroalgae are by far the better competitors on a coral reef and can outcompete hard corals for space. But the continuous grazing of macroalgae by herbivorous fishes and invertebrates allows crustose coral and algae and hard coral to dominate the substrate. Echinoids are among the group of most important benthic grazers in the tropical marine environments. Diadema antillarum, or the long-spined sea urchin, are found in tropical western Atlantic and can aggregate in high densities. Densities of diadema are inversely correlated with algal density. Simply put, more diadema means less algae. Less algae means less competition between algae and coral, and less competition means there is greater hard coral cover and more habitat for reef life. Acropora palmata, or elkhorn coral, and Acropora cervicornis, or staghorn coral, are branching coral that once dominated Florida Keys and Caribbean coral reefs. Studies have shown that areas with higher cover of Acropora are correlated with high levels of biodiversity. This may be due large in part to their growth mechanisms. These corals branch up from the substrate and provide a 3D habitat and structure, making the perfect habitat for many small reef fishes and invertebrates. In the 1970s, coral reefs in the Atlantic and Caribbean oceans began showing signs of coral decline. The decline has been attributed to a suite of stressors, including both natural and anthropogenic or human-caused stressors, including hurricanes, overfishing, climate change, rising sea surface temperatures, ocean acidification, and many others. Many of these events have been correlated with an increase in frequency of coral bleaching and coral disease events, which take a dramatic toll on the remaining coral populations. In the early 1980s, an unknown waterborne pathogen spread throughout the Caribbean populations of diadema. Diadema mortality spread from Panama throughout the Caribbean via ocean currents. The disease was first reported in Punta Galeta, near the entrance of the Panama Canal, and then reported for the rest of the Caribbean. Mortalities were preceded by tissue loss, spine loss, and lack of function in an urchin's tube feet, which resulted in the inability to attach to a substrate. The urchins were weakened to the point where they no longer hid in crevices during the day, but rather stayed out in the open on the reef. Urchins were picked off by predators, including grunts, parrotfish, and wrasses. Populations decreased considerably throughout the Caribbean. The diadema die-off is said to be one of the largest, if not the largest, and widespread marine mass mortality events ever recorded. Did you know? At Punta Galeta, the numbers of diadema changed from an estimated 14,000 individuals per hectare in June 1982 to 0.5 individuals per hectare in May 1983. The die-off of these urchins tipped the balance of the reef in regards to coral versus macroalgae abundances. As diadema began to disappear, reefs began to shift from coral-dominated reefs to macroalgae-dominated reefs. Even though reefs show signs of stress before the diadema die-off, it wasn't until after their mass die-off that the macroalgae abundances greatly increased and coral populations starkly declined. There is great evidence to suggest that this shift is something called a phase shift, where the dominant species in an environment shifts to another dominant species due to changes in that environment. In areas where diadema have died off and macroalgae is now dominant, scientists have been able to show that restocking these areas can significantly decrease the macroalgae on the substrate, allowing coral cover to slowly come back. These findings suggest that coral reefs are able to be restored back to their once coral-dominated phase. Did you know? Diadema were once so abundant on reefs that their removal was actually contemplated and planned. Tourists snorkeling on the reefs often stepped on them, and their presence was characterized as noxious. If they only knew then what we know now. Over 60 groups and organizations in more than 14 Caribbean countries have been identified as actively participating in acropora restoration programs throughout the Caribbean. These groups use a variety of methods to grow staghorn and elkhorn coral in offshore coral nurseries until the fragments reach maturity and are eventually outplanted onto coral reefs where they once existed. Projects are working to understand best practices for coral restoration and provide strategies for mitigating stressors that still affect our reefs. But what about the urchins? It is clear that their role and the role of other herbivores is of vital importance to coral reef health. Methods for diadema restoration are currently being investigated. Martin Moe began work on a diadema restoration project in 2001 to investigate techniques for culturing diadema from larvae to adult. The idea is to propagate enough urchins to relocate back to the reefs to increase urchin density. He has developed specialized culture vessels that can hold hundreds of thousands of urchin larvae. After several months, the larvae settle and metamorphosize into small juveniles and grow into adults. Moe has been successful in completing this process a few times and has a stock of adult urchins in his lab. However, there seems to be a critical point in urchin development at the metamorphosis stage, where some batches just don't make it through. Organizations such as the Moe Tropical Research Lab and the Florida Aquarium are working with Moe to replicate his work and understand how to overcome the critical point at metamorphosis. Once the code is cracked, urchins will be relocated to Florida Keys and Caribbean reefs, ramping up diadema populations at various reef sites. Increasing diadema densities would not only decrease the amount of macroalgae on the reef, but would also increase the number of urchins on the reef that can successfully spawn with one another, allowing the urchins to begin to come back naturally. So there is hope for our reefs after all. There are still many questions to be answered, but it seems possible that our reefs can be restored back to their coral-dominated states. There are additional factors that will need to be managed, but there are things we can do now to better understand the biology of coral reefs, to better protect and preserve them.